my name is Ester Lujan. I'm a senior at NMSU and my major is genetics. I work in Dr. Amanda Ashley's lab where we focus on investigating the role of various proteins involved in two of the DNA double-strand break repair pathways called non-homologous end joining and homologous recombination. A DNA double-strand break happens when there's a break in both of the DNA strands, and this type of DNA damage happens as a result of several stressors. For example, normal intracellular metabolism and ionizing radiation, and they're a serious threat to genome stability. Dr. Ashley's lab also focuses on investigating potential chemotherapeutic targets for a highly fatal type of breast cancer called triple-negative breast cancer. I became interested in working on this project after I learned that this type of cancer disproportionately affects African American and Latino women. My project focuses on investigating the recruitment of a DNA damage repair protein called DNA PKCS, together with another protein called gamma H2AX in the absence of another protein called NEC4. Overall, we want to see if there's any change in cell biology in response to DNA damage when cells are depleted of NEC4. DNA PK plays a role in joining together two broken DNA strands, specifically in the non-homologous end joining repair pathway. It does this by protecting the two broken ends after damage and activates other proteins to repair the break. DNA PK is a large hollow enzyme which is constructed of two parts, the Q7080 heterodimer and a catalytic subunit called DNA PKCS, which is the main focus of my project. NEC4 is a kinase, meaning that it takes part in a reaction called phosphorylation, where it adds phosphate groups to other proteins, potentially changing their activity. NEC4 may play a role in activating proteins that interact with DNA PKCS or with the protein itself. Another important protein for my project is called gamma H2AX. H2AX is a histone variant that, once it's been phosphorylated, is called gamma H2AX, and it serves as an indicator of DNA damage, specifically double-strand breaks. By assessing gamma H2AX, we can visualize where DNA damage is inside of the cell and what other proteins are present at the damage site. I will also observe proteins that interact with NEC4 in the formation or resolution of R loops. We will work on assessing genes in which R loops accumulate. So R loops are a DNA-RNA hybrid structure which is composed of two strands of DNA and one strand of RNA. An R loop can form when the replication fork, which is the area where DNA replication happens, runs into a transcription bubble. Once they accumulate, R loops can cause severe DNA damage and threaten genome stability. We are investigating if DNA PKCS plays a role in decreasing R loop abundance by detecting R loops in specific genes. We will also determine if NEC4 has a role in how R loops accumulate in a DNA PK dependent manner. Over the summer, I will learn new techniques and polish upon others I have already learned, such as mammalian cell culture. I will learn to conduct a DNA replication assay to visualize replication forks after treatment with a DNA damage-inducing drug in cells with normal or reduced levels of NEC4. I will conduct immunofluorescence and imaging to visualize the co-localization of DNA PKCS and gamma H2AX in the nucleus of BT20 triple-negative breast cancer cells and MO59K or J glioblastoma cells, which are isolated from a single patient, but one expresses DNA PKCS and the other doesn't. I will also learn how to treat cells with an siRNA to knock down NEC4. We hope the insights we gain through this research help us understand if NEC4 impacts how DNA PKCS is able to promote genome stability by facilitating accurate DNA repair and replication, both in the context of double-strand breaks and R loop accumulation.